This is part 2 of population genetics. This is the portion that we would have done in class on March 5th, but we're going to do instead through a recorded lecture. So this is going to be some review of a couple of the concepts from last time, as well as some more practice problems. I've posted this as a slide set as well. That way, if you want to try and work through the problems and try and delete the answers from the slides and figure it out on your own, you're welcome to do that too. As a reminder, population genetics focuses on two kind of main things that we're going to be calculating. One is the allele frequency, and the other is the genotype frequency. And we'll be looking at both of these as we go through the examples. Remember from last time, we have two equations. P plus Q equals 1. This equation looks at the allele frequencies with P standing for the dominant allele and Q standing for the recessive allele. And then we have P squared plus 2PQ plus Q squared equaling 1, which looks at the genotype frequencies. In allele and genotype frequencies, by convention, if there are two alleles at a locus, P and Q represent their frequencies. And the frequency of all alleles in a population therefore has to add up to 1, which means that P plus Q equals 1. And you may remember last time we talked about the Hardy-Weinberg equation. The Hardy-Weinberg equation predicts the allele and genotype frequencies will remain the same from generation to generation if a population is in equilibrium. And that Hardy-Weinberg equilibrium is that P squared plus 2PQ plus Q squared equaling 1, and also that P plus Q equals 1 is part of this too. Now, Hardy-Weinberg has 5 main components that need to be kind of true for it to hold in equilibrium. The first is no new mutations. The second is no natural selection. The third is that the population is so large that the allele frequencies do not change just because of random sampling error. The fourth is no migration, and the fifth is random mating. Last time I asked you a question, I'm going to ask you the same question again. Do you think we can find populations like this? Not necessarily. However, and actually the answer is no, not really. All of these things happen all the time. But we can still use this equilibrium to figure out whether or not things are really outside of equilibrium. Quiz time! What do the following stand for? P, Q, P squared, 2PQ, and Q squared. At this point, I want you to write down what you think each of these stands for. You're going to want to pause this until you've written it all down. Think you have your answers ready? Hopefully you've written them down. Here are the answers. P is the frequency of the dominant allele in the population. Q is the frequency of our recessive allele in the population. P squared is the proportion of the population that is homozygous for the first, that dominant allele. 2PQ is the proportion of the population that is heterozygous. And Q squared is the proportion of the population that is homozygous for that second, that recessive allele. Now we're going to move into some examples. This example we went through last time. This is the 4 o'clock plants, where the flowers come in red, pink, and white. And if you remember with the 4 o'clock plants, the red and the white are both alleles, but it doesn't have a clear dominant and recessive. When you have a heterozygote, it's actually pink because both red and white are dominant traits, so both are always going to be expressed. You are looking at a population of 4 o'clock plants in this example. There's 100 plants, and you count them. You find 49 are red, 42 are pink, and 9 are white. Okay, well then you're told to calculate the allele frequencies and the genotype frequencies for the white plants. And The equations are here on the screen, but I have a feeling that looking at this is really confusing for some of you. For some of you, it might make perfect sense, but here's what we're going to do. We're going to go through this example kind of again, and we're not going to look at the equations that are written here. We're going to do the math all on our own. Ready? So how did we come up with those answers on the last screen? Well, we know there's 100 4 o'clock plants. We know 49 of them are red, and the red ones have CR as their trait. The big C is standing for color. The R means red, whereas the W would mean white. We know that 42 of them are pink, so they're going to be CRCW because they have both a red and a white. And 9 of them are white, so they're going to be CWCW. So we have that information, and now we want to know the allele frequencies. So what are P and Q? And then the genotype frequencies, or what are P squared, 2PQ, and Q squared. Hopefully you remember P squared plus 2PQ equals Q squared. And here you can see that the P squared is going to be CRCR. So then P is going to be just CR. 2PQ is CRCW. And Q squared is CWCW, with Q then being just CW. Got it? Okay. So now we're going to take what we know and we're going to calculate genotype frequencies. You take the number of each genotype and you divide it by the total number. We can only do this because our heterozygote is pink. Generally speaking, a heterozygote looks just like a homozygous dominant plant. So in general, this won't work. And in the rest of our examples tonight, or today in this recording, this won't work. But because we're lucky here, this does. So how many are red? 49. So what percentage are red? 49 out of 100, or 0.49. So the frequency of red flowers is 0.49. That means P squared is 0.49. 2PQ would be our heterozygotes, our pink flowers. And we know that there were 42 pink out of 100 total flowers. So the frequency of pink is 0.42. And then the Q squared is the frequency of our white flowers. Well, we counted 9 out of 100 white flowers. 9 divided by 100 is 0.09. So we've been able to calculate P squared, 2PQ, and Q squared just by counting the frequency of each of our genotype, or each of our phenotypes, which are at this point in time our genotypes, for the flowers. But we're missing 2 things here. We don't have P or Q. They're just not there. So we're going to calculate them. So we can calculate now our allele frequencies. Because P is going to be the square root of P squared, and Q is going to be the square root of Q squared. So when we do P equals the square root of P squared, we discover that P equals the square root of 0.49. P is 0.7. Okay. Q is going to be the square root of Q squared. So it's the square root of 0.09. So Q is 0.3. But hopefully you're remembering that we already know that P plus Q equals 1. So let's check it. 0.7 is P, 0.3 is Q. 0.7 plus 0.3 is 1. So it's right. So we know that 70% of the alleles in the population are CR, that red allele, and 30% of them are CW, the white allele. Now we're going to try some new examples that we haven't seen before. For our first example here that's brand new, we have some shapes on the screen. Our shapes are pentagons. How many pentagons do you count total? And then how many do you count that are red? And how many do you count that are green? I'm going to give you a moment to count. Hopefully, you counted the same numbers I have, a total of 20 with 14 red and 6 green. Now we're going to use this information to calculate some frequencies. So we're going to assume that red is dominant, and we're going to say that since it's dominant, it's a big R, and that green is recessive, which is a little r. And we're going to assume this because there's way more red than there are green. So then, what are the frequencies of big R and little r? Or what are the frequencies really of P and Q? So in this example, our big R for the red allele is going to be P, and our little r is going to be Q for green. So this is kind of asking us for allele frequencies, isn't it? Yeah, it is. So how are we going to calculate this? Well, it's going to be hard to calculate for the big R because heterozygote dominant and heterozygotes. So the homozygous dominant and the heterozygotes are both going to look red right now. But we know the ones that are green, they're homozygous recessive. So let's calculate then what Q is. And we can calculate that. We know that we have 6 pentagons out of 20 total. So that tells us that Q squared equals 6 divided by 20, or Q squared equals 0.3. And then to calculate Q, we can take the square root of Q squared, or the square root of 0.3, which is 0.55. I'll give you kind of a moment there to do the math, but that's what we find that it is. And if we know that Q is 0.55, well, we know that P plus Q equals 1, so that means that P is going to equal 1 minus Q. So to calculate P, or the allele frequency of the letter R for the red allele, we would say 1 minus Q, or 1 minus 0.55, equals 0.45. So we've calculated our allele frequency of our dominant allele, the red allele, to be 0.45. And then the allele frequency of our green allele, our recessive allele, is 0.55. P equals 0.45, Q equals 0.55. Now we can go on to calculate some genotype frequencies. So then what are the frequencies of RR, RR, and RR? Or, as we look at this, what are the frequencies of homozygous dominant, heterozygous, and homozygous recessive? These are our genotype frequencies we're looking at now. And if you remember from that last slide, we discovered that Q was 0.55 and P was 0.45. Now we can answer this question because our Hardy-Weinberg equilibrium equation, that P squared plus 2PQ plus Q squared equals 1, that's how we get our genotype frequencies. So to get the number of homozygous dominant, big R, we just need to calculate P squared. So we know that P is 0.45. That means we're going to take 0.45 and square it. And our answer is going to be about 0.20. For the heterozygote, that big R, little r, that's represented by 2PQ. So we're going to do 2 times P times Q. 2 times 0.45 times 0.55. That equals 0.495, but we're going to round up to 0.5. And then how about for our homozygous recessive, little r, the greens? Well, it's just Q squared. So you take 0.55 and you square it, and you get 0.3. This means that 20% of our population is homozygous dominant, big R, big R. 50% are big R, little r, they're heterozygotes. And then 30% are homozygous recessive, little r, which if you went back to the last slide, you would discover that's what we found, that 30% of our population was green and 70% was red. So this is right. Hopefully you're catching on a bit. Don't worry, we still have a few more examples in case you're feeling like you don't quite know what's going on. Example 2. I'm going to make this one a little bit easier. This one's not really going to make you do any math, but it's going to make you think. You are studying an octopus population. And using the Hardy-Weinberg equation, you calculate the following frequencies for eye color. P squared equals 0.26, 2PQ equals 0.5, and Q squared equals 0.24. I didn't tell you anything about this octopus eye color or about the octopus population other than the only thing you looked at was their eye color. You don't actually need any more information about the population to look at this math. So I want you to look at this for a moment and think about it. You've got 3 things you need to answer here. What percentage of your octopus population is heterozygous for eye color? What percentage is homozygous recessive? And what percentage is homozygous dominant? You have everything you need on this slide. I'm going to be quiet for probably about 20 seconds to let you think about this. If you need longer than 20 seconds, you should pause it. Okay, I'm going to show you the answers. So if you haven't calculated them on your own yet, you want to pause it now. The answers. What percentage is heterozygous? This is asking you, what is 2PQ? That 2PQ, remember, stands for heterozygous. So the percentage of the population that's heterozygous is 2PQ, so it's 0.50. What percentage is homozygous recessive? Hopefully you remember that homozygous recessive is Q squared. So the percentage that's homozygous recessive is Q squared, which is 0.24. And then the last question, what percentage is homozygous dominant? Well, homozygous dominant is P squared. And if homozygous dominant is P squared, then it is 0.26. So you would say that 50% of our octopus population is heterozygous, 24% is recessive, and 26% is homozygous dominant. And that's exactly what we have here. We didn't need to do any math to calculate this. The answers were just right there in the question. Now we're going to do another example. In this one, your lab partner comes to you and they say, I didn't write down all the data. I was studying a population of frogs and, well, I don't know how to calculate genotype frequencies. All I have are the allele frequencies. And they tell you that they figured out that P was 0.75 and that Q was 0.25, but maybe they fell asleep or something and they forgot to get the rest of the data. And they're begging you, can you help me calculate the genotype frequencies? What do you think if you have P and Q Can you calculate the genotype frequencies? You can. The answer to this is, of course you can. P squared plus 2pq plus q squared equals 1. It's our Hardy-Weinberg equation, right? The p squared plus 2pq plus q squared equals 1. And you know p and q, so clearly we can calculate everything here. Now, if you're looking at this slide right now, you're going to discover this is a little different than the PowerPoint slides I had posted on Brightspace, which had a mistake in the math. Sorry about that. So I've gone back through and redone the math here. P squared would be 0.75 squared because we know that P is 0.75. So P squared equals 0.75 squared, which equals 0.5625. And for us, we're going to count that as 0.56 because you can't have part of a frog in this. Your frogs are homozygous dominant, heterozygous, or homozygous recessive, not like a half of something. So we discover that our homozygous dominant, or our p squared, represents 0.56, or 56% of our population. How about our heterozygotes? Our heterozygotes are that 2pq. So 2 times 0.75 times 0.25 equals 0.375. Or, if we round it up, 0.38. So 38% of our population is going to be a heterozygote. How about our homozygous recessives? That's going to be q squared. So 0.25 squared, which equals 0.0625, or for our purposes, 0.06. So we've discovered that 56% of our population is homozygous dominant, 38% is heterozygote, and a whole 6% is homozygous recessive. You were able to help out your lab partner and calculate everything because you knew P and Q. Now we're moving on to example 4. Examples 4 and 5 are very similar to example 1, but we're going to see if you can do the math again when we change things up on you. So how many total crabs, how many purple, and how many orange? It may look yellow on your screen, but on mine it's orange. So I'm going to let you do the counting. I'm going to give you about 20 seconds to count. If you aren't done counting, you should pause it now. It's a total of 30 crabs, 18 purple, and 12 orange. Okay, well this gives us some information. Now we're going to see if we can calculate our allele frequencies and our genotype frequencies from this. And I'm hoping that having these multiple examples are going to drive into your brains how to use these equations. If you are still really confused, come talk to me. But I can assure you, if you can do all 5 of these examples, you can do all of the math that is on the exam 3 for population genetics. What you're going to see on the exam is very much like at least one of these examples, maybe a couple of these examples. So if you can do math like in these examples, you're totally fine, okay? Now we're going to see if we can solve example 4. So let's assume that purple's dominant and orange is recessive. And so we don't have to use the letter P and confuse ourselves. I picked A for purple and orange. And because purple is dominant, it's a big A. Because orange is recessive, it's a little a. So then what are the frequencies of big A and little a, or P and Q for this population? This is asking for allele frequency. Remember, the letters just P and just Q, that's all about allele frequency. Well, we can calculate it just like we calculated back in example 1. There's 12 orange crabs out of 30 crabs total. So we know that the genotype frequency of orange is 12 divided by 30. That means that Q squared is 12 divided by 30. So 40% of our population is orange. So Q squared equals 0.40. Q is the square root of Q squared. So it's the square root of 0.4, which equals 0.63. And we know that P plus Q equals 1. So if P plus Q equals 1, P equals 1 minus Q. So P equals 1 minus Q, which means 1 minus 0.63. So P is 0.37. So the frequency of the allele for purple, that big A, is 0.37. And the frequency of the little a for orange is 0.63. So we've calculated our allele frequencies here. And once we know P and Q, we can go and calculate our genotype frequencies. So then what are our genotype frequencies? The big A is our homozygous dominant, the heterozygote, big A, little a, and then the homozygous recessive, little a for this population. These are our genotype frequencies again. And when you think genotype frequency, you need to think P squared plus 2PQ plus Q squared equals 1. So since we know P and Q, we can calculate everything else. So our homozygous dominant, that big A, that's going to be equal to P squared. So we take P, which is 0.37, and square it. And that tells us that our frequency is 0.14. For our heterozygote, we know that's going to be 2PQ. So we take 2 times 0.37 times 0.63, and that gives us 0.46. And then we do our homozygous recessive, little a. So that's going to be Q squared. So that's 0.63 squared, which when you do the math is 0.4. So this means that we've learned that 14% of our population is homozygous dominant, big A, which was what color? Does anyone remember? That was purple. 46% are heterozygotes, still purple, and 40% were orange, the homozygous recessive. Once again, if this is confusing, pause it, work through the math on your own, figure out if you can solve this. Go back and just look at the first slide of this where you were seeing the colors and see if you can work your way through the math. And we have our last example. I picked rabbits for Dr. Ficara in case you were interested, because Dr. Ficara has a pet rabbit. You might not know that, but she does. I have pet guinea pigs and I couldn't find a little guinea pig shape, so I picked the rabbits because I figured that was fun. And yes, if you're wondering, I picked crabs and octopus earlier because, well, I studied marine biology and I love it. So now we've got our last example, which is rabbits. I want you to count how many total rabbits, how many blue rabbits, and how many pink rabbits. I'm going to give you, this time I'm going to give you 30 seconds. If you haven't finished counting, you probably want to pause it. So our total number is 42. 33 blue and 9 pink. It's going to be more difficult this time around, isn't it? The numbers aren't going to be quite as even, but we can still calculate everything. So we're going to assume that blue is dominant. We're going to give it the big B for that. And we're going to assume that pink is recessive, so we'll do a little b, because there are a whole lot more blue rabbits than there were pink. So what are the frequencies of our different alleles? What's the frequency of the blue allele versus the frequency of the pink allele? Well, the blue allele, the big B, is going to be P. And the pink allele, that little b, is going to be Q. So this is an allele frequency question because we're asking about the frequency of a single letter, the alleles, that P plus Q equals 1. This is the same type of math we did in examples 1 and 4. We know there's 9 pink rabbits out of a total of 42 rabbits. So Q squared equals 9 divided by 42, or Q squared equals 0.12. And to calculate Q, we take the square root of Q squared. So Q equals the square root of Q squared, which is the square root of 0.21, which means Q equals 0.46. And we still know that P plus Q equals 1. So P equals 1 minus Q, or 1 minus 0.46. So P equals 0.54. This means that 54% of our alleles are blue and 46% are pink. With me so far? So now we're going to look at the frequencies of our homozygous dominant for blue, our heterozygotes, and our homozygous recessive for pink. Now we're looking at genotype frequencies. And when you think genotype frequencies, you better be thinking that P squared plus 2PQ plus Q squared equals 1. And remember, we just calculated P and Q, so we can plug those in and do the same calculations all over again here. Our homozygous dominant is P squared, so it's 0.54 squared, or 0.29. Our heterozygote is going to be 2 times P times Q. 2 times 0.54 times 0.46, which equals 0.50. And then our homozygous recessive is Q squared, so it's going to be 0.46 squared, which is 0.21. So 29% of our population has the genotype big B, big B. It's homozygous dominant. 50% is our heterozygote, big B, little b, and 21% is homozygous recessive, little b, little b. So we knew that most of them were blue, some of them were pink. This is the end of the recorded review for population genetics. This is a portion of our class from March 5th. And I'm going to ask at this point in time that if you have any questions about this, if you are confused about population genetics, email me, come talk to me, set up a time to meet with me, come to the review sessions. Please, please do that. Population genetics is not going to be a huge part of exam 3, but if you don't understand population genetics, then you might have some problems. And I'm going to say that not all of the questions on population genetics are going to be math-related questions. Some of them are going to be like questions on things that were in the last recorded lecture. So I hope you had a really great spring break and Monday off due to weather. All right, thanks for watching.